And last but not least, our third speaker in the lectures is Professor Sylvain Baillet. Sylvain is the Tier 1 Canada Research Chair of Neural Dynamics of Brain Systems and has been Associate Dean of Research of McGill's Faculty of Medicine and Health Sciences since 2019. He's also a founding member of the Standing Committee on Science of the Canadian Institute of Health Research and was elected chair of the Organization for Human Brain Mapping in September 2023. Congratulations for that.
Thank you.
He joined the Montreal Neurological Institute in 2011, where he founded an extremely successful MEG core program. In fact, the MEG unit he founded at McGill has become the second busiest scanner on campus after the 3D MRI. So Sylvain, maybe you can share your wisdom here, how you did all that.
We don't have a train passing by the building.
And among his many achievements, Sylvain co-founded the Open MEG Archives in 2015. That's the first open access repository of MEG data. And together with Richard Leahy in USC, who is my PhD advisor actually, and John Moser in UT Houston, his team develops the Brainstorm software, an international NIH project which has grown a community of more than 43,000 registered users and 3,000 related publications. So I actually know Sylvain for over 20 years now, and I've been collaborating on this project as well. And Sylvain's lab contributes to advancing our understanding of the large-scale network brain activity, specifically how it enables complex behavior and how it is altered in disease. And his lecture today is titled Systems Neuroplasticity. I hope it hasn't changed, right? Systems Neuroplasticity of Predictive Mechanisms in Human Perception and Cognition. Or something.
I don't remember putting plasticity in there, but that's okay. That's okay.
Neurophysiology. Apologies.
Thank you so much, Dimitrios, and thank you everyone for sticking around. You can imagine myself as a giant cookie because this is what's going to happen after my talk, coffees and cookies. Very grateful, very good to be back. I think last time I was speaking here was for the inauguration of the MEG Center here. So thank you very much again for having me this time around. And thank you, McGinn, for sponsoring and for the support. So I'm also very grateful for the fact that both previous speakers have touched in depth at very fundamental aspects. I will try to cover with a slightly different angle, very complementary, I hope, in terms of feedback, feed-forward, top-down, bottom-up mechanisms. But the framing of my talk today is to look into the complexity of human neurophysiology as captured by MEG, to better understand the predictive mechanisms of brain systems and how basically we're going to try to see together how basically perceiving is essentially predicting or modeling reality and to accounting for possible mismatches and learning from those mismatches. So without further ado, I'm going to look at the brain as a predictive machine. So this is not a new framework by far. This is really, aligned with a very long tradition in philosophy and neuroscience about looking again at how internal representation, how basically the human experience is a phenomenon that can be reported and observed by oneself, but also by others. That's exactly what we do in the lab, I guess. But the idea is indeed to look at the brain as a set of hierarchical networks. We're going to talk about primary network layers in the network. These networks project to higher order brain regions or deeper layers, like Leila mentioned many times and brilliantly today. These connections could be direct or they could be through different relays. And there are several relays, important relays or hubs in the brain, as you know very well. Could be cortical, could be subcortical, but that's the idea. This is just a concept here. And what is remarkable in the brain networks is that there's a great deal of feedback reentries, especially back to primary regions, but possibly also higher up in the hierarchy. And we're going to see what we believe is the essential role, basically, of these feedback connections to instantiate prediction at every single moment in time, so that maybe we can better explain why, basically, even perceiving things like social interactions can be resolved in an astounding amount of time, less than 200 milliseconds, from what we just heard. So depending on where you're coming from, could be neuroscience, could be engineering, could be linguistics, etc. We're going to look at the blue highways as the bottom-up highways and the green highways as the top-down with respect to the hierarchy of the brain networks. What we're going to posit as a hypothesis is that these top-down connections, they are, again, they enable, basically, the channeling of these contextual predictions based on the context, the environment, like me speaking in front of you today. This is in a certain spatio-temporal context. And basically, what we're going to look at is at the primary layers or early layers in the network as basically differentiators that's going to constantly compute a difference or a mismatch, potentially big or small, between these predictions that are issued internally and the actual external inputs. And this is an important, relatively simplistic, but very important concept, because you can imagine that depending on your goal in a certain context, you may want to adapt your behavior to achieve a certain or obtain a certain reward or achieve your goal. And if your internal representations are not good enough because you miss your goal or you don't get the award or reward you were looking for, you're going to have to update basically those internal models so that next time you're getting better. And that's the idea of learning. So from these very simplistic views of hierarchical brain networks, we can start digging into more conceptual or deeper concepts of brain and behavior. What I would like also to emphasize today is the fact that the brain doesn't live in a void; it lives in a body, and this is often, not. It's a bit of an oversight on behalf of neuroscientists, and me included, to believe that the brain again lives by itself, and maybe it's connected to the world through our senses, but for the rest of the story, we don't consider that the rest of the body is relevant. Although it's extremely important, of course, for many obvious reasons. The brain is highly connected to the gut. There are more connections to the gut between the brain and the gut than in the spinal cord, for instance. So many pathways that are increasingly studied both in health and disease. We know that heartbeats modulate attention, for instance. There is the gut-brain axis in many hypotheses relevant to chronic neurological diseases like Alzheimer's and mental health conditions. And of course, so there is the brain in the body and the body interacting with the rest of the world. So this embodiment basically is also very important in terms of imposing on the brain and therefore on brain computations, which are extremely expensive from a metabolic point of view, a limited budget that the brain has to observe, maybe not from millisecond to millisecond, but over time there is a fairly hard constraint that is imposed on brain computation, and we need to keep that in mind to test when we look at data, especially those that are time resolved at the natural scale of behavior, just like those we pick up with MEG, for instance. We can look at consciousness. This is not the topic of today's talk, but as an experience, like I said before. It's the experience we all have of us being embodied. It's us in our body interacting with the world. That's one possible definition of consciousness. So again, a long tradition of philosophers and neuroscientists looking at this. So let's try to be a bit more specific now. So we're going to look at context, the brain in a context, so brain in a body in a certain context. And we're going to imagine we are bird watchers and we are standing by a tree. And the possible outcomes is whether we're going to see a bird, take off and fly away from that tree, for instance. And because we are expert bird watchers, we expect certain species of birds, certain, patterns of flight probably, in a given environment, maybe here in Boston, in a park. And imagine that we are fairly certain of our knowledge, and we are in this environment with one single tree. So we're going to expect one bird of a certain type taking off and flying away from that tree. So let's imagine that we are in a context of relatively low uncertainty. So that means that we're going to give more weight to our internal representations and possibly less to our primary input regions. And we're going to considerably, bombard constantly our primary regions with these internal representations and predictions of where and when possibly the target might be manifesting. So we might allocate less budget to the primary encoding of the visual input by just anticipating from moment to moment, even more and more in time, as time goes on, maybe, that there is an event of interest that's going to happen. And this is the actual outcome. And let's say this is a very high, low surprising outcome, because this is the bird we were expecting in the only tree I'm looking at. So this is low surprise. So I'm going to give more weight eventually, update my internal representations by, reinforcing the model I based my detection of the target on by just anticipating there is maybe a nest in that tree, and I'm going to expect more of these birds taking off next time. So now let's move to an actual outcome that is completely at odds of what we would expect. In the field of possibilities, that's a very low probability that a car would actually, take off from that tree, except if we are maybe in Back to the Future. But anyways, so that means that we are facing a very high surprise outcome. So very low expectations of that outcome. That means that the error signal is going to be huge, and we're going to have to, next time, rely less on our internal model that there is a bird systematically taking off of that tree. And that's also part of the learning process to, next time, probably put more weight on our primary inputs and less weight on our anticipations from internal representations. So you can look at also an environment where uncertainty is much higher, where you see all the possible outcomes have almost equivalent probability. So let's say that instead of one tree, we have many different trees. And in a high uncertainty environment, you want probably to give more weight, less weight to your internal representations, because you have too many trees to look at, possible targets, even though you expect birds to take off from each of those trees, and give more weight or tune up the gain of your sensory perception, primary input layers, so that you give yourself more chances to detect changes in your visual field. That could be a bird or something else. And indeed, if you have the expected outcome, then you can update your internal model that there is indeed a nest in that tree you were looking at. And you can reinforce that model. But if you come up with a high surprise outcome, then again, massive predictive error will manifest, and you will have to update your internal model, and next time rely less on it. Okay? So sorry for those of you who know those things better than I do, and who've seen that many, many times, and probably in more sophisticated terms, but I thought I would set the stage to now go into the possible mechanisms that we might be able to observe with neurophysiology and MEG in particular, and the rest of the talk is actually a set of mechanistic hypotheses and some empirical evidence we have accumulated in the lab over the past decade or so that look directly into those things. So we're going to look at basically a possible relay or possible junction between primary regions, which we will simplify by saying they are essentially sensory regions, and the higher order regions in the cortical or the brain hierarchy in general. And one important relay is basically the sensory motor cortex. But also, if you look at the more current view of the hierarchy, the functional hierarchy of the brain, you have these blue regions that are essentially unimodal or acquainted to sensory processes versus those that are transmodal or integrative, and they don't specifically align along, there's not a clean border along the central sulcus only, but there are many others where possibly the representation of the body even away from motor actions, can actually make sense conceptually, but also neurophysiologically. So we're going to look into this. So in other words, we're going to look at non-motor functions or functions that relate the body to the brain and vice versa, and look at basically for today at the role of frontal motor circuits in non-motor functions and how they might play a role in perception without overt movements, for instance. So in terms of mechanisms, we're going to look at different neurophysiological pathways or the implementation of this boxology, if you will, representation of predictive brain processes. So we're going to look at exactly the same brain, but this time instead of boxes, we're going to look at circuits that are made of excitatory and inhibitory cells. And what we're going to claim is that when you have such elementary circuits, they're going to tend to oscillate. And they're going to tend to oscillate at the human brain within a range that we've seen earlier today in the two previous talks, between slow fluctuations around delta theta up to the alpha rhythm that is so prominent in the human brain. And nested within those cycles of slow oscillations, we're going to look at cross-frequency coupling with gamma activity, so faster frequencies. And we have such circuits pretty much everywhere in the cortex and possibly elsewhere as well. And we have those interactions between these different cells, these different circuits. And what's missing from the picture in terms of canonical frequency from electrophysiology, as you may have guessed already, it's the beta band, which is this intermediate frequency between roughly 15 and 35, 40 hertz in the human brain. And the hypothesis we are making, and we are not the only ones, I'm thinking about Pascal Fries and others, is to look at the beta activity, essentially in the shape and form of bursting of discrete events, as possibly being vehicles for top-down modulations or top-down signaling from internal models back to primary regions. So if we look now at the intimacy of the time series within one of those circuits, we're going to look at the red curve, at the red slow oscillation, as a proxy for the inhibition, if you will, of the net inhibition of the circuit. Again, these things have a metabolic cost that is not insignificant by far, so they have to go through these, phasic reinstatement of inhibition versus excitation by construction, by design, if I may say so. So again, this is just a zoomed view of this phase amplitude coupling between the fast burst of gamma and the slow oscillation up to alpha. So, and now we're going to look at an input to that circuit, and whether this input is predicted or unpredicted, we claim that it's going to change the phase of the oscillation. Why? Because probably the circuit wants to be able to encode the input with the maximum precision, both in terms of the features of the actual input, as we just saw from Leila, but also its timing with respect to a certain context. Think of the bird taking off at a certain moment. You want to be ready for that event. And if you can predict when it's going to happen, you're going to be better at detecting it at lesser metabolic cost, for instance. So if the first instance of input is actually unpredicted, maybe the circuit will be able to detect it and encode it and relay the information further downstream. Maybe not. But what's going to happen is that if there is an error signal that is actually issued locally, what one of the many things it's going to do is actually create a phase resetting of the red oscillation based on internal representations of the current context, and try to guess when the next input's going to happen. So if the next one is actually probably properly predicted, well, ideally you want it to fall into the burst of the gamma for reasons I'm going to try to explain in a moment. And we think that at least in a time domain, this time resetting, if you will, phase resetting of the slow oscillation will be issued or driven by beta bursts that are generated further higher in the brain hierarchy. So what's happening, and I hope I'll have time to show you some empirical evidence of this. What's happening at the level of the fast gamma burst? Well, if there is actually an input that is happening at a certain moment here, it's going to fall into one of these slots of the oscillation, which is actually a further decrease of the net inhibition at that moment. therefore, the input will be registered with higher accuracy and with lesser metabolic cost. And you can imagine that you may have multiple successive events within a gamma burst, and it might be a way to actually order in time the succession of events from the same sensory modality or different modality, depending on where you are in the brain network, so that you can actually make sense, basically, again, of your environment and refresh your model about the context. So this is very much aligned, for instance, with Ole Jensen's work. And we published a paper a couple of years ago that show evidence of that with MEG. And again, I hope I'm going to have time to take you through this. So now, enough talk about boxes and oscillations drawn with PowerPoint. Let's have a look at some data, finally. So again, I'm pretty obsessive about a few things. MEG is one of them. And when I talk about MEG, I like to talk about cross-frequency coupling. So looking at what the brain spends a lot of energy generating all the time. So if these different things coexist, they must live together by definition and work together possibly. And one of these things are these slow and fast oscillations. So we are looking at evidence of cross-frequency coupling, interdependence of these fast and slow rhythms. And actually, it's been demonstrated about, is it already 18 years ago? But not so long ago by Bob Knight and Ryan Canolty in Berkeley, where they showed with an intracranial recording in an epilepsy patient evidence, again, of cross-frequency coupling during resting state over the prefrontal cortex of that patient. And you see a nice alternation of high and low gamma power with respect to the phase of an underlying slow oscillation in the theta range for this example. And with Esther Florin a few years ago, we actually replicated this observation, but this time non-invasively across the whole cortex during the resting state of human participants. And everywhere we looked, pretty much, we found evidence of cross-frequency coupling in the human cortex. And then with Esther, we designed basically a possible model that would explain how we could detect basically brain networks in the resting state based on the statistics, the time statistics of phase amplitude coupling, in the sense that a brain network could be defined by the fact that it's a set of brain regions that align basically the phase of their net inhibition excitation, so that basically they work together and are excited together at the same time versus inhibited together at the same time. And then also show a greater than chance, if you will, level of coherence in the gamma bursting over time. So this is how we define basically that the blue or dark blue and red two regions would be connected in the resting state through phase amplitude coupling versus the green one, which is out of phase, which slow oscillation is out of phase, or the black one, because the gamma bursting is not coherent with the other two. So in a way, it's an extension of the communication through coherence framework proposed by Pascal Fries. So we did that. We looked statistically at the different modes of connections with phase amplitude coupling, and we were able to retrieve with MEG, non-invasively, the resting state networks, or things that look very much like the resting state networks that have now been, very much replicated and studied with fMRI, for instance. But the cool thing with MEG is that you can track also the fluctuations of this phase amplitude coupling, and you can demonstrate that these networks are not rigidly formed and shaped. They actually reshape over time. And so there is this idea of dynamic functional connectivity, of course. And when you look at the spectrum of these fluctuations, they peak at a frequency that is ultra slow, that is actually very much compatible with observations of those fluctuations with fMRI. This is a little bit of a side note. So maybe in the room there are people who've heard about oscillations, who are actually experts of oscillations, but also may question their functional relevance. And because, again, the mechanisms I'm trying to put forward in terms of how the hierarchy of brain processes is implemented in the brain relies on brain oscillations, I thought I would cite recent work we did with Philippe Albouy, who was a postdoc in the lab, who's now a prof at University Laval in Quebec City. and where we showed basically the causal role of brain oscillations in brain function. And the function that we used as a test model was working memory, auditory working memory. So Philippe presented short 3-note melodies to the participants in the MEG, and then there was a manipulation instruction, 1, 2, 3, 2, 1, 3, etc. The numbers relate to the notes in the melody. So here you don't have to manipulate the note during a retention period or maintenance period of a few seconds. And then you are presented with a test melody also of 3 notes. Could be the same, could be different, could be matching the manipulated melody or not. But you have to detect or ascertain and decide whether the middle note matches that is expected from the manipulation. It's very hard. I fail miserably as a participant myself, but the goal is to have difficulty in there so that we have a range of performances, and I will try to explain why. Because when you do the source mapping and you look at this retention period where people manipulate in their mind the short 3-note melody, you see an overexpression of theta activity. So this is time, and the dashed box represent the manipulation, the 2 seconds of manipulations in working memory. And you see a great peak in theta over the intraparietal sulcus during that period of time. And actually, when you look at the relation with behavior, there is a nice relationship. The more theta you generate, it looks like the better you are at the task. But this is just a relation. It's not a causal relationship. So to test causality, what Philippe did is bring back the participants on another day and actually deliver 5 pulses of TMS, repetitive TMS at 5 hertz, so every 200 milliseconds, over 1 second. And over the IPS, we verified with simultaneous EEG that indeed it would induce in the target region an increase of theta, and indeed it did. And then we looked at the behavior. We had the participants, after each delivery of the TMS, have a go at a working memory trial. And then when you look across trials, with the rhythmic TMS, you have a substantial improvement in the working memory with performance with respect to baseline in pretty much every participant, which you don't see if you deliver the same amount of energy to the brain, 5 pulses in 1 second, but in an arrhythmic fashion. So that was an important control to do. And you see you're back to baseline in that respect. And actually now the missing piece is that you want to relate this observation to the fact that whenever you deliver TMS over the IPS and there is an increase in working memory performance, there is also increase in theta activity with respect to baseline in that region, and that's also what we found. So that's a causal relationship between oscillations and behavior in that model of working memory. Actually, what we saw from David earlier today with speech and language is based on another fantastic feature that is a fantastic tool, I mean, whenever you want to study the brain with MEG or electrophysiology in general, but MEG in particular, is that whenever you present those 3 notes in a rhythmic fashion, or even more than that, like a full music score, as we saw, or even a speech, natural speech, you will end up entraining the brain at certain features, rhythmic features of the physical stimulus. So here, this is a study we did with Robert Zatorre and Emily Coffey, who was a postdoc with us, and who's now a professor at Concordia University in Montreal. So what Emily did is basically look at the frequency following response, which is a bread and butter or workhorse of audiology, where you present basically phonemes that are repeated over time. And this is the auditory envelope of the phoneme. And basically what you can see with MEG, if you look at the auditory cortex, is that there's a beautiful response that is in sync with the phoneme that is presented. But it's not only in sync, it's also, it has some energy, endogenous energy in there as an oscillator, because after you stop the delivery of the auditory stimulus, the brain circuits locally continue to be untrained. So that was really cool to see. And actually, you can even track in time the frequency content of that oscillation. And if the resting state oscillation locally is around 80 hertz, and you start delivering an auditory input that is modulated at 100 hertz, you're going to see the instantaneous frequency catch up with the presentation and eventually oscillate itself at 100 hertz. It's a little bit of a segue with respect to my talk today, but I thought I would mention this tool. This feature, because this is a really, really cool aspect that you can study with MEG, not only in the cortex, but also in the deeper regions of the brain. And here, for instance, we looked at the brainstem, and I don't think you can go much deeper in the brain than in the brainstem, and this is the auditory response of the cochlear nucleus, of course, after thousands of trials, so it's a very specific, framing of the experiment. But there is enough sensitivity with MEG so that you can go and detect those deeper regions, provided you give yourself enough signal on top of the noise. So I'm mentioning entrainment for several reasons. The first one is that we replicated, remember, the TMS study with working memory, establishing a link between theta oscillation, causally enhancing working memory. So Philippe, again, a couple of years after that, substituted the TMS with a rotating figure, a rotating 3D object that was presented a few seconds before each working memory trial instead of the TMS. And why rotating figures? Because they are known to untrain the dorsal visual pathway and therefore untrain potentially the IPS, which is the target brain region we were talking about, we were actually looking at to enhance working memory. So what we did is basically, first of all, we behaviorally, we replicated the observation that whenever you do that with a rotating figure object at 5 hertz, flickering at 5 hertz, you would have enhancement of the working memory performance. Versus no steam, or even showing also something that flickers at 5 hertz, but without a coherent rotation of the object. So that's really the visual 3D representation of the object that entrains properly the whole dorsal pathway. And when we looked at the electrophysiological counterpart, this time with MEG, because with TMS we had to resort to EEG after delivering the TMS. But this time we did everything in the MEG. And we found not only increase of theta activity, but also increased connectivity with the typical frontoparietal network that is engaged in most working memory paradigms, including this one. And again, the more you enhanced, you entrained the dorsal pathway, the more you actually improve the working memory performance. Again, now this time using only entrainment, not with TMS exogenously, but endogenously with, so to speak, with visual stimulation. So let's bring back the model, you remember, of the bottom-up versus top-down with the role of beta oscillations. Now I'm going to go more in detail into this observation, into this hypothesis of beta oscillations and bursting actually helping, predicting or channeling these predictions, especially in time, of when a salient stimulus, for instance, or like a target is expected. Here the targets are not the birds, they are these notes in the middle of a sequence of 5 notes. And to frame the experiment, we actually presented a target at the 4th position in the middle of this 5-note melody. It's actually not a melody, it's like a 5-tone, pure tone sequence, always the same, except that the 4th tone might be, every once in a while, a deviant in terms of pitch. So it's kind of an oddball paradigm, except that it is framed into a 5-note sequence. And actually, it's more like the 4 first notes that are very important because to the participants, because we know that they know that they will expect that the target is going to be in the 4th position in the sequence. So let's have a look at, the beta oscillations around that 4th note. So our predictions were that there would be, according to our model, some form of entrainment, first of all, of at least the auditory cortex at the 3 hertz frequency, which is the rate of presentation of the notes in the sequence. And on top of these oscillations, we would expect either gamma burst, but also beta burst that would be actually occurring at certain phases of the slow oscillation, just to make sure that as a signature of when the target is actually expected, so that the detection is optimal. So we have both this slow oscillation and this burst of beta that we expect to be, again, stronger when the target is expected, so that the detection is optimized. So this is what we observed. You may forget about the amusics, but this is a study with, in collaboration with Professor Isabelle Peretz, who's a cognitive neuroscientist who is an expert in music perception, music processing. So we tested amusics who cannot detect melodies, but also healthy controls. I won't have time today to unpack the results for amusics, but what this graph is showing, 0 second is the onset of the target tone, so that's the fourth in the sequence. And the y-axis represents basically the amount of phase amplitude coupling between the slow oscillation at 3 Hertz and the beta bursts that are detected locally at each single trial of the presentation in the auditory cortex. And what you see, we're using a time-resolved phase amplitude coupling method we developed with Sohail Assami, who was a grad student in the lab, is that there is indeed an increase, a marked increase over time in the phase amplitude coupling, culminating at the presentation time of the target. And actually, if you look at each single trial, so to speak, if you look at the density of the beta bursting, I hope I can convince you that the histogram of the beta bursting is actually very, very sharp with respect to what's happening afterwards, but even beforehand, before the presentation of the target, it's very sharp around the presentation of the target as we expected. It's building up gradually as we approach with the first note, the second note, and the third note, but it peaks at the fourth note, and then goes back to baseline levels for the fifth note. So the idea is that, indeed, we detected those bursts in the auditory cortex, but when we looked at the whole cortex map, this is just a synoptic cartoon that summarizes the results, we saw that they were actually possibly issued from the motor cortex, these beta bursts, and directed towards the auditory cortex, but also other areas of the ventral auditory pathway, like the IFG, inferior frontal gyrus. And this reminded us of a previous study we did with Benjamin Morillon, who was a postdoc in the lab and now is a PI in Marseille, in France, where we looked at temporal attention, and it was a bit of a more challenging task for the participants. They were trying to find a 1.5 Hertz sequence of tones in the middle of distractors that generated overall a 3 Hertz sequence as well. And we saw that there was this phase amplitude coupling around the 3 Hertz and the beta burst that were generated in the sensory motor cortex that were related to the processing and to the performance of the task. So again, motor engagement and activation of beta bursting and top-down prediction possibly on behalf of the motor cortex without overt movements. There was the participants did not move at all during the performance. And actually, when we looked at resting state, we realized that spontaneously there is an expression of beta activity, bursting activity that is directed between the regions that we looked at for that study, but possibly through the rest of the brain as well. So it looks like it's a mechanism that is, or at least a phenomenon that is present and that contributes to the temporal prediction, at least of events of interest in a certain context for here auditory perception and maybe visual perception as well. So I'm going to skip that. This is just, I'm going to finish with natural speech from pure sequences of pure tone to short melodies to speech. We're going to look at entrainment with greater and greater complexity and look at predictive mechanisms, so bottom-up versus top-down. David explained what speech entrainment or entrainment of brain circuits by speech is about. So we're going to look at some work we did with Peter Danhauser, who's actually now a postdoc with David Poppel in Frankfurt. He was a grad student with us. So again, we presented natural speech, actually TED Talks, to our participants while they were sitting in the MEG. And we looked at every input bit. So the bits we looked at are essentially each and every phoneme, each and every word. And of course, each and every word and phoneme are in a certain context, right? And this is what we care about. This is whether we can find in the brain a modulation of language circuits and language circuits activity related to context. So related to the fact that maybe we are in a very uncertain environment, so a certain noun or certain word, is very unlikely to be pronounced. So if I say lion now, it was probably not too likely to be pronounced in this context. So again, you recognize maybe the different landscapes I was showing a moment ago with the bird watching thought experiment, but this time this is for real. And we're going to look at surprise. So ideally, you want to track in real time. This is my point here. You want to track, if you want to look at predictive processes of speech processing in the human brain, you need to be able to look at least a form of proxy of uncertainty for each and every word, each and every phoneme, and surprise once these phonemes and words have been pronounced and uttered in a given context. But the problem is that, because, yes, let me just finish here. If you were able to have those, traces, which are potentially very correlated, right? If you are very uncertain about your context, you are very likely to be surprised. But even more so if you are certain about a certain context, and if a different word than you expected is uttered, you're going to be even more surprised. But if you had these things available to you in real time, you could use some form of regression with the MEG data, and you could look at basically, like Leila showed, a form of variance partition where you could look at what's the contribution of uncertainty to the brain response versus surprise. And if there is a contribution, if uncertainty modulates the processing of your of the auditory inputs, which is here natural language, then this is a demonstration that there are predictive processes, ongoing predictive processes during natural speech perception. So the problem is that in every individual, those variables, measures, are not observable. These are really internal, because they are attached to the internal representation of somebody's mental state or even history, if you will, with respect to what the story is telling. So that was before LLMs and before ChatGPT. Even though we published this only 4 years ago, ChatGPT was not available, or actually we were not aware that there might be a prototype available. But what Peter came up with is to actually train an artificial system on the corpus of TED Talks that we used, so that basically we could have proxy probabilities we could derive uncertainty and surprise from long stretches of language, so tokens of different lengths, and use those numbers actually in our regression model with brain activity. So that's a summary of what we used. We used actually LSTMs, which sound today, very outdated, but they did the work. They did the job. We used the transcription of 1,500 TED Talks, about 10,500 unique words. And we trained those LSTM systems so that, again, they predict the next phoneme and the next word in a sentence. And then we use those things as regressors. And what we found is that there are essentially there are multiple brain regions that are involved in the processes, but we use temporal response functions, which is a form of decoding of the response of brain circuits to each and every element of interest. So it could be a word or it could be a phoneme. And so what you see here in time, at 0, this is the word onset in the stream of natural speech. So this is not event related to each word presentation. This is extracted from ongoing brain activity without averaging, using temporal response function identification, which is a form of machine learning identifications of patterns that are recurrently present in the brain responses around certain events. But you see that they look very much like event-related potentials to single words, for instance. So far, so good. This is a form of validation of the approach, so to speak. And those patterns were projected actually preferentially around the primary auditory cortex. And then Peter found another major dimension in the data that projected actually very proximally to the primary auditory cortex, but a bit more, posterior and a bit more anterior, so around the primary auditory cortex. We found something, and this is a detailed view, we found a temporal response function that was very different, that was much slower, because probably we're looking at something that is deeper in the hierarchy. So remember, I'm trying to follow my train of thought, that we're looking at the blue region at the top, which is more primary, and the green region at the bottom, which is more deeper in the hierarchy of the brain circuits. Another cool thing, I think, with the tracking of uncertainty and surprise in real time is that you can really look at how these canonical temporal response function, actually empirical, I should say, TRFs that you extract from the whole corpus of your data are modulated by the variables of interest. So uncertainty, effect of context, and surprise, effect of your updating your internal representation based on your context, your previous model, but also based on your on the actual information that you get from the stimulus. And what we found is actually in a context that is highly uncertain, as I was trying to explain in my intro slides, there is an increased response, early response to the words or phonemes that are being presented. So if you are uncertain about what's going to happen next in the speech flow, what's going to be presented, you're going to pay more attention potentially. I'm using these words with quotation marks. We haven't tested attention, but it's very likely that the gain, if you will, the encoding of the incoming words is increased, and that yields a sharper response function to the word in a context of high uncertainty. And we observed this enhancement of the early response only in the primary auditory region, which is where the words are supposed to be encoded. Whereas when there was an effect of surprise, when actually there is a greater surprise, you see that there is an effect, but only in the later stage of the responses and in both areas, which could be a trace of what I was trying to explain at the beginning, which is maybe a manifestation of a later update of your internal model based on the fact that you have not potentially predicted the incoming word or phoneme precisely because of the uncertainty and because of the potentially noise in the flow of information. I think I should stop around here. So I will try to show you I will finish with something we are pretty excited about that has a more, if you will, pragmatic viewpoint on those things. So this is the idea that basically if our brains function at a certain level, a certain scale that is observable with MEG, well, if you look at how these different oscillation rhythms and dynamics of the brain are expressed over time, well, there could be a signature of yourself. There could be, something quite unique to your personality or to your person, I should say. Why? Because they represent or they encapsulate how you encode information and how you predict information, right? Depending on your environment. So we revisited the notion of brain fingerprinting that was pioneered by Emily Finn and her colleagues with fMRI, but this time with MEG. And this is the work of Jason da Silva Castanheira, who has just graduated from his PhD last week. And so the idea is that you're going to take 2 snapshots of brain activity with MEG, and you're going to look at basically how they differ in time for a certain individual. So let me explain that. We used and leveraged the Omega database, but now we extended our observations to the CAMCAN database with hundreds of participants. Here we were a bit around 150 participants. The snapshot of brain activity we took were 200 days on average apart, so it's almost 8 months apart. We looked at durations of brain activity that were quite short with respect to fMRI, or very short, 2 minutes, and we actually pushed the envelope down to 30 seconds. So the idea would be that you get a snapshot of the brain activity of someone during 30 seconds, and you try to match that person again 200 days from now with, again, another snapshot of 30 seconds, and you hope to, if this is really a matter of brain fingerprinting, you should be able to differentiate that person from the crowd based on these fingerprints. So what we did is basically brain imaging, extracting the power spectrum and the connectome from these data. So it's task-free data, just resting state. We actually looked at both connectome and power spectrum and ended up using only power spectrum because it was sufficient. But anyway, you have a bunch of participants and you have a confusion matrix, basically, which is a correlation between each participant and themselves and the others. And you hope to see a nice diagonal between the different participants. So what we found is that first we tested within a session, so not 200 days apart, but a few minutes apart, whether we were able to basically differentiate between individuals. And you see that we are approaching perfect accuracy, maybe 95% accuracy, using both connectome or either connectome, I should say, or spectral activity, and across the frequency bands of interest. My favorite is beta because of the reasons I was trying to explain. I'm trying to frame this idea around the fact that beta activity is a signature of your internal representations of the world. So probably this is the frequency band that carries the most information about yourself, but that's a conjecture, we'll see. And then we looked at the between session, 200 days apart, and you see that mind the scale, it's very different. It's from 75 to 100% this time, and you see that we are still above 90%, and still very high, including with the beta band. And then we looked at basically 30 seconds of data, and we were still able to differentiate between individuals 200 days apart with a very short snippets of data. And then we looked at which brain regions were the most salient in the fingerprint for each frequency band, and it looks like the most global distribution of salient regions is expressed in the beta band. And we replicated this in Parkinson's patients. Now we are trying to take this and translate this tool, basically it's just a tool, into things that could be fun for neuroscience, but also useful potentially to clinical neuroscience. And we replicated these observations with Parkinson's disease patients, and we're able to better explain the differences between the healthy controls and the patients, and we're even able to decode the disease stage from the brain fingerprint of patients. So I should stop here. I will just finish by thanking my lab, of course, and thanking you all for your patience and attention. I'm looking forward to our discussion. Thank you so much.